In this video, I want to show you how both T lymphocytes and B lymphocytes coordinate to form this specific immune system and destroy pathogens and those cells that are infected with pathogens. If you have not yet watched my two videos on how B lymphocytes and T lymphocytes are clonally selected and then clonally expanded, please do so first. The first thing that happens, as if you've watched the other two videos, is clonal selection, where antigen presenting cells in the lymphatic system will find the complementary receptor on the B lymphocyte, which is an antibody, and the complementary receptor on the T lymphocyte, and those particular B lymphocytes and T lymphocytes and no others are now selected. Once selected, they undergo rapid mitosis, which is called clonal expansion, and they differentiate into various subsets of these lymphocytes: B memory cells, T killer cells, T memory cells, T helper cells, and B plasma cells. Now, the B plasma cells will not release antibodies until a second signal. That signal comes from the T helper cells, which release chemicals called cytokines. Those cytokines will stimulate the B plasma cells to continue dividing and differentiating and go on to release lots of antibodies. Those antibodies will circulate in the blood and they will bind to complementary fitting antigens, be it on pathogens or also infected cells. The T killer cells will seek out, by binding with receptors on the cell membranes of infected cells, and destroy those infected cells by releasing a cocktail of harsh chemicals, hydrogen peroxide, hydrolytic enzymes, for example. Don't forget the T and B memory cells will remain in high number inside your body lymph nodes in preparation if you ever encounter the pathogen once again. Put this all together, the humoral response involves the production of the antibodies. So the antibodies in circulation will bind to complementary fitting antigens, such as these that are on the pathogens. What they do then, we'll discuss in another lesson. They may simply stick the pathogens together, called agglutination, or even destroy the pathogens directly. Now, this is all very well and good, but if you have infected body cells, viruses, for example, inside these body cells will continue to burst and infect other cells. So this requires a cell-mediated response, where you have T killer cells which will circulate in the body, the blood, in your tissues, and they will recognize the complementary antigens on infected body cells and release the hydrolytic enzymes, hydrogen peroxide, harsh chemicals to destroy these cells. People describe the specific immune response as being a pincer movement, because you have the T lymphocytes destroying infected cells and you have the antibodies from the B plasma cells binding to the antigens on pathogens. It is a specific immune response because the other cells of the body are not touched. So only those cells or pathogens bearing the complementary antigen will trigger this immune response.